Biology. How do enzymes work? What are enzymes? Well, they're biological catalysts. In other words, proteins which speed up chemical reactions in the cell. And these are proteins twisted into a globular shape. Without enzymes, living organisms wouldn't be able to survive, as the chemical reactions needed to survive would happen too slowly. Well, first there's the substrate, the substance on which the enzyme acts on. Then, the location where the substrate and the enzyme bind is called the active site. This is where the reaction will occur. When they bind, it forms an enzyme-substrate complex. Once they bind, the chemical bonds in the substrate will weaken, causing them to separate, in this case. The previous type of reaction, which involves the breakdown of large molecules, polymers, into its building blocks, monomers, is called hydrolysis. The opposite reaction, which involves building or synthesizing polymers by joining together monomers, is called dehydration synthesis. After the reaction is done, the enzyme can be used over and over again. The following are examples of enzymes, like carbohydrates, which breaks down polysaccharides into monosaccharides, the building blocks of carbohydrates. Protease breaks down proteins into amino acids. Lipase breaks down lipids into its building blocks, which would be glycerol plus fatty acids. There are factors that affect the speed of the reaction, like the area, temperature, pressure, concentration, and pH. To make the speed faster, you would have to increase the area, increase the temperature, increase the pressure, increase the concentration, and make the pH ideal. Enzymes are very specific, as only one type of substrate can fit into their active site. This allows the control of biochemical reactions, without which the body would go haywire. When the optimum temperature is surpassed or the enzyme is exposed to extreme pH conditions, it denatures. This means its shape irreversibly changes and it cannot function correctly anymore. Because its shape changes, the substrate can't fit in and it can't work. Optimum temperature is a balance between denaturing, and it takes advantage of the fact that at a higher temperature, particles move faster and the reaction can happen faster. Humans used to get a lot more enzymes from their diet. In the past, raw foods helped resupply their digestive tract with beneficial enzymes, and nowadays they are cooked or heavily processed. Both deplete natural enzymes. Most raw foods nowadays are refrigerated, which lowers the natural enzyme count. Thanks for watching.